Welcome back to Lab at Home with the Museum of Life and Science. My name is Peregrine, and I am so excited to be doing the program with you all today. I hope that all of you will help contribute to the program and help lead it with your thoughts and questions and ideas in the moderated chat box so that we all can have a part in what we are learning today. We are going to be talking about DNA today. So before we get started with our experiment, I thought maybe we could talk about DNA and what it is and what it does. I would like to ask all of you, if you know anything about DNA, can you share it in the chat? This is great. One of my friends was saying it comes from your mom and dad. Yeah, your DNA is something you inherit from your parents. So we have DNA. That's an interesting idea. We have another friend saying that DNA is in cells and stuff. I agree. DNA is in cells. It is in the cells of all living things or recently living things. I have another friend saying it's a part of the body. Yeah, so our bodies have DNA in them because our bodies are made out of cells. All of our cells, almost all of our cells, have DNA in them. And the cells of all things that are living, or I should say recently alive, also have DNA inside of them. So DNA is kind of like, it's kind of like a recipe. I like to say it's a recipe for how to make you or me, or more specifically to make certain parts of me. Like I have a recipe for making my skin cells, or I have a recipe for making my heart or liver cells. Right? We said all living things have this. In order for things to grow, they need to have DNA to tell them how. So we know what DNA does. What exactly is it? I don't know if you all always envision the same picture that I do. This picture right here. Something like this is often what I imagine when I think about what DNA is. I imagine the shape. Oh, we even had a friend saying a little ladder. Yeah, the shape is very ladder-like. It's kind of twisty. We see those little rungs on those twists. What's going to be called our helix. It's a double helix even, that kind of cool twisty ladder. And it's made out of molecules, just like all the rest of the things in our cells. The full name of DNA is deoxyribonucleic acid, which lets us know what kinds of proteins it's made out of. So DNA is a type of molecule called a polymer. A polymer, that's something like, there's lots of different kinds of polymers. Some are living, some are not. If you've ever made slime before, slime is an example of a polymer. A polymer is just a type of molecule that likes to chain together. So when you make slime, it's something that's very stretchy because it likes to hang on to itself. It chains up. DNA is the same way. That's how we get that shape of them. What I would love to do with you as an experiment today, talking about DNA, is I would like to take some DNA out of some cells so that we can look at it. Now, we were just saying that DNA is a molecule. It's inside of our cells. How are we going to get it out, and how are we going to see it? So inside of our cells, there's so much DNA, right? It's coiled so tightly because it is a polymer. There's so much DNA that if we could get it out of all of our cells and look at it, there would be enough of it for us to see it with our naked eye. So what we need to do is extract the DNA from a bunch of cells, and there should be enough of it for us to see. Okay, so we could extract our own DNA. A lot of times that's done using a cheek cell. You take a little cheek cell swab from the inside and you can extract the DNA from that. But I think I'm going to use something else. And I asked all of you to grab some supplies to help you extract DNA from something else. I'm going to be doing a classic one today. I'm going to be extracting DNA from a strawberry. There's a special reason why a strawberry, a couple of different reasons, which we'll talk about in just a minute. But first, I'm going to switch to experiment cam so we can talk a little bit about what supplies we need for today's experiment. So we've got a bunch of different supplies that we can use for today's experiment, the first thing you'll need is something to get DNA out of. We recommended a bit of, we recommended a plant specimen. We specifically recommended something like a fruit because fruit is soft and it's easy to squish up, which is going to be a big part of today's experiment. But I have a strawberry. I'd love to know if any of you are doing the experiment along with us. Can you share what you're using? I've used lots of different things before. I've used strawberries, of course. I've used bananas. Kiwis are another really good one. Ooh, we have a friend who's using grapes. That'll be a cool one. Oh, I'm excited to see that. I'm excited to hear about that. Ooh, we have another friend who's using bananas. That's another great choice because they're so mushable. So we have our plant specimen. They're living things, so they have cells, so they have DNA inside of their cells. Oh, I have another one who's using tomatoes. That's so interesting. I can't wait to hear about tomato DNA extraction. Some other stuff we're going to need for our experiment is we're going to need some water. And I have my water in a little cup here. You should have yours in something that you can mix a solution into. So just a little cup will do fine. You're going to need some salt also. You're going to also need a little bit of dish soap. And you're going to need a cup, a little plastic bag, and something like a fork. I'm using a fork. I also have a little pair of crafting tweezers here. You could also use a toothpick, something like that. We're going to be needing to use something like that. And we also will need some isopropyl alcohol. So this is rubbing alcohol, and you can see I have 91%, which is a very high percentage of alcohol. You can use any percentage kind of that's higher than 60% is going to be useful for you, but the higher the better. And I've done something else. You might even have been able to see I have these little kind of freezer rods for water bottles, and they're helping me to keep it cold because cold isopropyl alcohol is going to be really useful for this experiment. So cold isopropyl alcohol. And then my strainer. If you don't have a little one like this, you can use a different type of like mesh here. You can also use a coffee filter or even a paper towel. And speaking of, I always try to keep something to wipe up messes whenever I'm dealing with a bunch of different stuff. There are some other ingredients that we said were optional. We'll probably get to those later, but I'll at least mention them now. We were saying if you're going to be using a certain type of plant specimen, a mortar and pestle would be useful. So it's not necessary if you have something soft like a strawberry or a banana or a tomato, but it could be helpful depending on what you're using. And we asked you to grab some types of some products that have enzymes in them. So we have a meat tenderizer here. You can buy that in the grocery store. And we have some 100% pineapple juice. So these are not needed for our experiment, but I will be checking them out a little later on so we can see what effect they have. We've got our bunch of different supplies. Let's get to our experiment for the day. So we need some water, and I'm going to need you to grab your salt. And I'm going to stir mine up to help it dissolve. This is not warm water, so it may not dissolve all the way, but I think it will dissolve as much as I need. So I went ahead and I put my salt in here, and I'm stirring it up to make some salty water because the salt is going to help me break down those cell walls. It's nice and abrasive, and it's something that's going to help break open the cell wall, kind of like breaking open an egg, and then inside will be that DNA. And I'm going to add something else to my mixture here. I'm going to add a little bit of dish soap. So not a ton, I'm not dumping a ton in there, but a good blob of dish soap into our solution here. And I'm going to continue to stir it. I've got my salt in here, and I've got this dish soap. Now, dish soap is going to be really helpful in breaking down those cells because these cells have an outer layer called a lipid layer, right? So lipid is fat. So the soap is really good at breaking through fat. So it'll break down those lipid layers and help free the DNA from the cells. So the salt is going to help abrade the cells. And the dish soap is going to help kind of melt away that lipid layer to release our DNA. Let's go ahead and kind of combine our different elements here. So we have our DNA freeing solution here, and I want you to grab your plastic bag. This is going to be our kind of chamber where a lot of our experiment is taking place here. And I want you to take whatever it is that you're going to split up, whatever plant sample you have, and I want you to put it inside of here. And I put one strawberry in there just for fun. I might add one more strawberry. However much you have. I did go ahead and take the leaf off of it just because I won't be able to squish up the leaf with my hands. But if we've got a nice squishy sample like a strawberry or a banana or something like that, we'll be able to do it with our hands. So I have my, my strawberries here. What I'm going to do is I'm going to carefully pour a little bit of my DNA freeing solution into my bag, like that. So this is about the amount that I have. This is probably even more than enough. Once you've poured it, what I want you to do is carefully zip your bag up. So this is something we want to be really careful about because we're going to need to start squishing in a moment, and we don't want any of our solution to leak. There we go. So my zipper is good and zipped. Now comes the fun part. We need to start squishing up our plant specimen. I need to start squishing up my strawberry so that we can start to free the DNA out of those cells. So if you're doing it along with me, let's go ahead and start. We're going to start just squishing it with our hands like this. So I am just squeezing them until they pop open like that. And ideally, what is going to work the best for us is that we don't have any big pieces left. So if you're like me and you can't really squeeze anymore, you can put it down on the table and you can start to press it. This is somewhere where our fork can help us again. Now you can probably just use your hands, but if you need to, you can also use your fork to help squish it. You just want to be careful that you don't accidentally pop a hole in your bag. Something else I should have done that would make it easier is making sure there's not too much air in my bag. So I'm going to squeeze the air out of my bag. There we go, and reseal it really carefully. Okay, and now there's a little less air to contend with, so I won't be, I can just mush around the solution that I have. So if you're doing this along with me, I'd love to hear how it's going in the chat. Is what you have easy to squish? How's it going for you? Are you finding out that you're making a good looking plant smoothie, plant and salt and water and salt and dish soap smoothie? We have one friend who says it's going pretty well. That's good. That sounds good. Oh, so you used a banana and it is nice and squishy. That's great. A banana is a great option. I was realizing that these strawberries that I got may not be super, super ripe, so they might be a little bit harder, and that's okay. I think we can still get what we need from them. All right. So I don't know about you, but I've got a good looking smoothie going on here. Not one I exactly want to drink, but we have created a mixture here where we freed the DNA from the different cells, right? And all of the DNA and all of the little bits and pieces are floating in this bag, but we need to separate the DNA from all of the rest of it. We want to be able to see that DNA. Now, what we're going to need to do first is get all the clumpy bits of pulp or whatever big chunky parts are left from your plant sample. And this is where our strainer and cup will come in. We're going to take this and we're going to open just the corner of our bag. And I'm going to really carefully pour it in to my cup. So what this is doing is it's straining out all the clumpy materials so that I can hopefully get mostly DNA. So all of the stuff in here, we do not need any of this stuff. This can go in my rinse bowl. I'll go ahead and put that in my rinse bowl here so it's nice and clean. Now in here, in my strawberry soap salt DNA lemonade, is all of the DNA floating around. But I want to be able to see that DNA. We said earlier that DNA is a polymer, that it likes to link onto itself. We're going to add one last ingredient here to help all of those DNA molecules link up and make it so that we can see clearly where the DNA is. So you're going to take your rubbing alcohol. Again, this is my 91%, and it's pretty cold. I was keeping it in the freezer. It's still pretty cold. And we're going to pour our alcohol into our cup. There's not a set amount. I like to pour at least a quarter of the amount of stuff poured in here, or maybe you could say at least a half of the amount that you already have poured in here. And the way that you're going to pour it is you're going to carefully pour down the side. Or if you have a spout like me, you can carefully pour in the spout. What we're doing when we put the alcohol in here is the alcohol is going to help us push our mixture into different layers. Right? It's going to kind of help push those molecules we were talking about all together. Now, as you are adding your alcohol, if you're doing this along with us, tell me what you're noticing. I'm going to hold it up to the camera to see if any of what I am seeing will show up. Is a little bit of separation happening. Oh, interesting. I have a friend saying that there is a little bit of color change happening on mine. Yeah, I think it does look like the color changed a little bit here. I'm definitely seeing some color change here. I almost have a little layer of clear substance on the top of my solution, and it looks like there's some bubbles inside. I'm going to hold this carefully up to my camera. Those bubbles were not there before, right? These bubbles are the layer of our DNA. So what I want you to do is I want you to take your fork or your tweezers or your toothpick or whatever you have, and I want you to try and lift out with your fork. I want you to try to lift out what you have in there. Whoa. So check out what I have on my fork. I don't know about you, but to me this looks a little gross. It looks like boogers to me. This stuff that's all sticky and stringy is DNA. This is the DNA from the strawberry that I just squished up. We freed the DNA, it came out of the cell, and here it is, all really good at clinging to itself. Now, depending on the different amounts that you put of stuff, your different ratios of salt or dish soap or the type of plant that you used, you may be seeing more or less. I'd love to hear if you're doing this along with us. Did you get anything that looks kind of like this? This is really interesting. So I have got this beautiful amount of DNA here. I'm going to place it onto my Petri dish. Now it's okay to touch. I know it seems kind of gross to touch because you're like, ew, it's a booger. But remember that this is just strawberry and water and salt and soap. So here is our DNA. This is the DNA of the strawberry. Remember, none of the pulp got through our strainer. So this is just the DNA that has clumped up here. I'm going to try one last part of our experiment here. So earlier in the program, we mentioned two other ingredients that we might be able to use in our experiment. Those were some pineapple juice or some meat tenderizing powder. So these two ingredients have one thing in common. They both contain the enzyme called bromelain, and pineapple juice contains it naturally. Bromelain is a type of enzyme. An enzyme is a chemical that helps break things down. So I have a question for all of you. Do you think that if we added some enzyme, some bromelain enzyme into our plant breaking down process, do you think we might be able to yield more DNA? What do you think? What's your hypothesis? Do you think that if we have something that helps us break down cells, that we might be able to get more DNA? I think we should find out. So I'm going to replicate our experiment from earlier where we had 2 strawberries. So I'll go ahead, here's 1 strawberry. Here's 2 strawberries. per bag, and I would love to be able to try both. I want to see which of these enzymes does a little bit better. So I'll have 2 strawberries in one bag, and then I'll grab one more, and I'll put 2 strawberries in this bag too. Take the tops off, because I won't be able to squish those up. Okay, just like before, what we're going to want to do is put them inside of our plastic bags. 2 for this one, and 2 for this one. And we are going to take, just like before, we're going to take some of our dish soap and salt liquid here, and we're going to pour a little bit of it in. I think that was about more or less as much as the other one. All right. Let's add a little bit of our meat tenderizer powder to this one. So this one is the same as before, except we're going to add a little bit of this meat tenderizer. So I added some of it in. I just wanted to have you check out those ingredients there. It says that there's some salt added, there's some dextrose, but here that third ingredient is that bromelain enzyme, and a few other things, probably some anti-caking agents to keep it powdery. So even though this has a lot of salt in it, that's not a new ingredient, right? We still have salt inside of our DNA extracting liquid. So it shouldn't change our results too much. It might just be like the equivalent of adding a little bit of extra salt here, but mostly we're trying to measure the effects of the powder. So I'm making sure that that powder is nice and dissolved, and it's a pretty fine powder, so it does a pretty good job of dissolving. Now I'm going to push all the air out of my bag and zip it closed. And before we get to squishing, I'm going to take my other bag and I'm going to replicate the experiment, but instead of tenderizer powder, I am going to use some 100% pineapple juice. So I'll put some of this in here. That look more or less the same as before? All right, so I'm just going to give it a little bit of a glug here. Ready? Glug, like that. Okay, so then go ahead and do the same thing. Push as much of the air out of your bag as you can and zip it up like that. Now, we have 2 different, wow, look at those color differences. That's so interesting. So we have 2 different experiments here, each of them using different forms of the bromelain enzyme. I'm going to go ahead and squish these up. Just like before, we're going to squish these up, start to release the DNA from those cells, and see which one was able to extract more DNA. All right, so I have crushed up my 2 different bags. The one in my hand here is the pineapple juice, whereas this one is the meat tenderizer. So just like with our other experiment, we're going to go ahead and pour them out into these 2 different cups. I'm going to be sure to pour the same amount as in the other one, which was about 70 milliliters of each. So this one is my pineapple juice. Put my pineapple juice near it. And I will have my other cup help me hold my strainer there. And pour. Go rinse this in my rinse bowl. All right, nice and clean. Now, likewise, I will do my meat tenderizer, which is in our second cup here. All right, so now we have our 2 different solutions here, about 70 milliliters of each. And we're going to go ahead and add our isopropyl alcohol. So I'm going to go ahead and we're going to add it to both, kind of right on their spout here. I'll try and add approximately the same amount. I'm watching my fill line here. Move this gently so that I can fill this one. Our experiment was to see which one of these types of bromelain enzyme, right, in the pineapple juice naturally occurring or in the meat tenderizer powder, which one helped us extract our strawberry DNA a little bit better. I'm giving it just a minute to start to collect on the top. I'll show you all there. That's looking pretty good. You can kind of see that foamy stuff, almost looks like spit. That is our DNA here at the top. This one is our meat tenderizer. This one is our pineapple. I'm going to take my fork and we will compare the amount of DNA in each of them. So here, whoa, is from my meat tenderizer. Pretty good. Go ahead and just use my finger there. That's pretty good. And now I'll try with the pineapple. That's looking pretty good too. Let me grab this a little bit more over here. All right, so let me bring these little clumps of DNA a little bit closer to our camera here. This is really interesting. Here was our original yield. I've seen much bigger yields of strawberry DNA when you use strawberries that have been frozen and thawed, and I think that's because they're less hard than a fresh strawberry, so their cell walls have already been broken down. So I've seen yields that are much larger than this, but for the purposes of our experiment, we saw this was our original yield here. It's dried up a little bit, but that's okay. We can still see how much is in there. And then here is the yield from the pineapple juice and the meat tenderizer. I don't know, I think it's close, but to me it looks like the pineapple juice may have done a little bit better. That is so interesting. My guess, my thought would be, is that maybe there is more bromelain enzyme in our pineapple. Now, I guess it's worth considering that pineapples are also living things with cells and their own DNA. So the question is, okay, wait, did this one have a higher yield because we put more cells in it? And that's an interesting thought. I think we should be considering that for sure. My response to that would be the amount of pineapple cells inside of this juice are probably less than the amount of cells in this strawberry, because the strawberry has a bunch of, it has the pulp, it has the seeds, it has all the different parts which are made up of cells. And my guess would be that it's not like we added another strawberry's worth of pineapple DNA. If there's pineapple DNA present in our yield, I would say it's only a little bit. Plus, I'll say I don't really know the effect of bromelain on its own cells. On cells from its own plant. I don't know, I'll have to look that one up. Really, really cool stuff though. I really appreciated the opportunity to do this experiment with all of you today to get our different yields of DNA. It was so interesting to see the effect of an enzyme on our extraction process. And I don't know about you, but I am going to be experimenting with a bunch of different living things. There's so many opportunities to extract DNA because there's cells all around us, as long as it's kind of easily squishable, or if maybe it's easily squishable when we use a mortar and pestle. We could use different kinds of fruits and vegetables. If you wanted to use your mortar pestle, you could use stuff like leaves, flower petals. I was even thinking I might try some dry active yeast, because even though this is dormant, yeast is still a living thing. It's a fungus. So I wondered if I could get any DNA out of yeast. I'll have to try that one afterward. Friends, I hope that you will continue to experiment as I will. Thank you so much for joining us today. Yeah, thank you so much, my friends. Yeah, thank you too. All right, I hope we'll see you next time for Little Lab at Home. But for now, have a great rest of your day.